encourage you to interrupt me. Let's keep it informal. I'm keeping this at a high level overview. Haven't really provided a whole lot of specifics for OGT, but we could certainly discuss that as well, about what some of our ideas are for applying pluripotent stem cells. So again, thank you all. I'm fortunate to work with a lot of the rare disease community. I'm a member of a few different medical advisory boards, and I really enjoy this type of outreach and discussion, because it really helps direct us and keep us grounded in really the important things we need to do on the research side. So in terms of what my laboratory does is we focus on two different models to study rare disease. One are genetically engineered mice, and the other are human-induced pluripotent stem cells. Let's put my laser pointer on so it makes it easier. And so, the focus we'll do today are on human-induced pluripotent stem cells, or iPS cells, or iPSCs. So I'll use some of that jargon. And why do we use these models in a research laboratory? Well, the reality is, for so many rare diseases, we don't understand how the disease progresses and what are the complex manifestations associated with the disease. And that is in part because it's rare, but also because it's difficult to do these types of studies in humans. And so this is why these models, which we call preclinical models, are so useful. And importantly, and I'll give an example of how we are using them ultimately for therapeutic discovery. Ultimately, we want to be able to find viable, strategies to identify therapies for these rare diseases. And so we'll focus today on human iPS cells. So I do want to, look back and start talking about the relative advantages of some of the other different models as well. And so, everyone here are very familiar with rare diseases and, and why we need to study them. And so particularly with children, because I'm in a children's hospital, we're very keen on rare diseases because half of all individuals with rare disease are children. And the biggest challenge is, although we have had tremendous progress in diagnosing rare diseases in terms of genome sequencing and genomic testing, the reality is even after diagnosis, 95% of all rare diseases do not have a therapy. And so these are the things we need to focus on. So understand these diseases better, and by doing so, identify new potential therapies. And this is what we do. And so human pluripotent stem cells are a really valuable model for us to study rare disease. And so I want to go through just some real basics about what are pluripotent stem cells, and in particular, what are induced pluripotent stem cells. Well, or I'll call them iPS cells, for our jargon. And again, please interrupt as I go along if you have any questions. So what are iPS cells? Well, they're a special cell type we can grow in the lab in a dish, and we can grow them indefinitely, and they're a little fickle to work with, but we can grow them in the laboratory. And importantly, they are actually generated from human cells, so we can make human-induced pluripotent stem cells. And really, they have several remarkable features about them. They're remarkably similar to embryonic stem cells, except they're not from the embryo. So all the controversy of embryonic stem cells, put that aside. These are not embryo-derived cells. In fact, these are actually cells that we can, generate from patient cells very readily, and I'll talk about that in a minute. If you look at them under the microscope, this is a picture I took of some of our iPS cells that we have growing in the laboratory. They're kind of a boring-looking cell type under the microscope. They got a big nucleus and a tiny little cytoplasm. They grow in these little clumps, but their rather boring nature, at least visually, is hides some of the remarkable, properties that make them so valuable. So how do we actually get in iPS cells, from a patient? Well, we could take cells from an individual, and we'll call these somatic cells, and under a particular, process, which I won't go into detail, we can convert those somatic cells to induced pluripotent stem cells. And those are the cells that have many of these very primitive features of the early embryo. What are somatic cells? So we could take cells from an individual. Somatic cells are just any cell type in your body that's not a sperm or an egg. And so, oftentimes when we make iPS cells, want to generate iPS cells from a particular individual, We would either get skin cells, and we could get that from a skin biopsy. More frequently, because skin biopsies can be a little bit uncomfortable, we've improved things so we can now use blood cells. So a simple blood draw. So these are cells from an individual that we can then take, and these are live cells that we modify through reprogramming to make iPS cells. And we can do that fairly readily, and the process is really quite efficient in the laboratory. Any questions? Otherwise, I'll keep going. And the key thing is these are all patient derived. So I'll talk about why that's so powerful. And so that includes individuals with a disorder. We're now capturing the genetic material, a genetic disorder. The genetic material, which includes that variant that causes the disease, is present in the somatic cells. So they'll also be present in the induced pluripotent stem cell. And that's a really powerful tool. We now have cells in a dish that came from a single individual that we can study to understand the disease. Okay?
Mm-hmm.
So that's pretty cool. The other really, really amazing thing, really the critical thing about induced pluripotent stem cells is they have, that's what pluripotent means, that they can turn into other cell types. And in fact, we could take these cells, and under the right conditions, we can push them in a dish to turn into other cell types. And then these are cell types that look a lot like the cell types that are present in our body. So one example is we could push the cells to become heart cells or blood cells or cells that make insulin or the cells of the brain. And so I'll talk about why we think that is so powerful and important when we study disease. Importantly, and I'm not going to talk a whole lot about this, but we also have the ability to form our organoids. And so what these are is we could take the induced pluripotent stem cells, make them form an aggregate, a clump of cells, and under the right conditions, we can push them to differentiate into something that looks a little bit like a very primitive organ in the body. In particular, brain organoids or cortical organoids are a very powerful tool to study neurological disorders. And so you probably have seen organoids and their power and their utility. It's the capacity to study more complex tissues that are derived from these induced pluripotent stem cells. Sometimes I don't like this, but some people will call these brain organoids mini brains. They're not mini brains. They are tissues that look in some ways like a very primitive brain tissue. They lack a lot of things, but I do want to bring that up. So it's this capacity for us to turn these iPS cells into other cell types that we think are relevant to the disease are really what allow us to leverage them to study different disease biologies. Okay, and so over the years we've made a lot of different iPS cells, and we've differentiated them to a lot of different cell types. So these are some microscopy images. These are of a spinal cord neuron lineage that we had developed. So it's a protocol to turn the iPS cells into something that looks like cells from the spinal cord. We're very interested in blood cells for some of our disorders. We can very efficiently make blood progenitors. This is a differentiation. I can't do that. Sorry, I want to show this because it's kind of cool. If I can. This is actually a video of some cells that we have in a dish. I think if you look closely, you can see the cells are moving. You can see that they're beating. Can I convince you of that? Because we differentiated these cells to cardiomyocytes, under the microscope, they're actually beating, and they're forming a functional, beating cardiomyocyte layer of cells. So this gives you an idea of some of the things we could turn our cells into to study different diseases. So why do we want to turn these iPS cells into more specialized cell types? In other words, why do we want to differentiate them? Well, you can imagine, because we could try to study diseases in the iPS cells. We can even try to study the diseases in the skin fibroblasts. But in reality, if we're studying a neurological disorder, we really want to be studying the neurons of the brain that we think are possibly defective. Similarly, if we work on a pediatric disorder, which they have heart failure, we believe we need to study the heart cells to really understand that. And the reasoning behind that is these specialized cells that are in our body have specialized functions. So heart cells do one thing your entire lives, and they do it very well until we have a disease, in which case that can be problematic. And importantly, if there are genetic disorders that give rise to cellular defects in these specialized cells, we need to study that. And I would argue that diseases manifest themselves very differently depending on the cell type that we want to study. So in other words, for example, if we want to study OGT deficits associated with OGT conditions, we want to study, we believe that these biochemical and molecular defects would be different in a neuron compared to a heart cell, compared to a skin cell. And so that's the reason why we really want to study these in these specialized cell types that are relevant to the disorder that we're looking at. And the key thing is this is all done in a dish. So we can add things back, we can modify the conditions, we can do things. It's a very manipulable system for us to do experimental manipulations. Does that all make sense in terms of why this differentiation procedure is so, critical and relevant for us to study disease?
Sure.
Okay, great.
Mm-hmm.
So that's an overview of how we can generate pluripotent stem cells from a patient.
Sorry, I think Miranda has a question. Miranda, do you want to unmute yourself and ask the question?
Sure. I was just curious, are these the same kind of cells like they use for cancer treatment? My son had to have 3 stem cell transplants a long time ago, but it was like back then it was explained that it was used to just give him his entire immune system back. Are these the same kind of cells?
It's a great question. There are many other stem cells in our body. The stem cell transplant you had was a bone marrow hematopoietic stem cell transplant. So our blood cell progenitors live in our bones. And so what they did is they pulled some of those blood progenitors out from one individual and transplanted them into your child. In this case, these are a different type of stem cell. And in this case, induced pluripotent stem cells actually do not exist in the body. And so these are an experimentally generated model, but they are physically and biochemically very similar to some very, very early cells that did exist in the embryo. So these are different stem cells. So it's a very good question for asking that. Does that answer your question?
Completely answers it. Thank you so much.
Great. Thank you. Okay. So, and please, I really welcome any questions you might have. So we've talked about how we can take cells from a patient or an individual and make iPS cells from them and use them to study disease. We can also take advantage of the fact that another remarkable property of pluripotent stem cells is that we can genetically manipulate them. And this is a major activity in my laboratory where you probably have heard of CRISPR or CRISPR-Cas9. These are the very specialized tools that have become developed in the past 15 years or so that allow us to make genetic modifications in a variety of scenarios or contexts, including pluripotent stem cells. And so I won't go into a lot of detail, but we can design specific tailored CRISPR genome editing tools that can allow us to target very specific regions of the genome. And we can make genetic alterations using these CRISPR reagents. Experimentally, that is advantageous because it allows us to add back pieces of DNA if we want to do that experimentally. In the clinic, it's also being explored as a potential for repairing. So if we can go in and repair a defective gene, CRISPR has that capacity. And there's been some very high-profile advances in this very recently that's shown the feasibility of this for some genetic disorder. So CRISPR-Cas9 is a remarkably important tool for us, both in the laboratory and it's emerging as a potential therapeutic. There's a long ways to go for therapeutics, but there is remarkable progress of this as well. So why do I bring this up? Well, I'll give a couple examples of why, how we use CRISPR in our laboratory. But one other way where it's particularly important is we can use CRISPR to generate a disease model rather than using patient cells with the disorder to make the iPS cells. So how do we do that? Well, we can take induced pluripotent stem cells from an individual that doesn't have a disease, as much as we know. So these are quote unquote, I don't like to use the word normal, but these are iPS cells from someone who does not have any known disease. With the right CRISPR tools, we can go into those cells, make genetic modification, including making a mutation in a gene that we're interested in that can cause disease. And so this is a means for us to generate genetically altered cells that can recapitulate or mimic the mutations or the pathogenic variants that may be identified in patient genome sequencing. So this is a really valuable tool for a couple of reasons experimentally. This allows us to make a large number, potentially, of different iPS cell lines that we grow in a dish, and each cell line has a different mutation associated with a disease. And we can do these fairly quickly, and we don't have to capture cells from individuals with a disorder. We can instead genetically engineer the cells to have the mutation. And that actually allows us to accelerate certain experiments quite a bit. The other real advantage of this is experimentally, the only difference between this original cell type from this unaffected individual to when we make this genetic modification with CRISPR, the only difference is this genetic modification that we've created. So these cells are otherwise genetically identical. And that actually turns out to be a very helpful tool for us in our experimental procedures. We have a comparison, and we can basically say whatever biochemical or molecular deficits we see, or cellular deficits we see in these mutated cells, compared to the the original cell line. That's a very powerful experimental tool. If we have a large panel of patient-derived cells, we can do comparisons, but because every individual is very different, and every pathogenic variant is potentially very different, there's a lot of experimental noise with that. And so this experimentally does allow us to get past that. So there's a lot of advantages to using CRISPR to actually generate these disease models in a dish.
Hey, Jay, I think there's a couple questions that people have their hands raised.
And I apologize. It's a little hard for me to track things down, but thank you for interrupting me. So what do we got?
So my daughter has the genetic defect. So obviously she has two X genes in her body. She's using the one that has the defect. The question is there a way with CRISPR to change her to the other X that maybe doesn't have the defect that, could help her, coming from a very layman's sort of perspective?
Yeah. Conceptually, that's an interesting application of CRISPR. Operationally, that would be a really challenging experiment to do, but I believe it would be feasible. I probably have to defer to someone who knows more about X and activation than I do, because the way it works, and I think you're probably familiar with this, is females have two X chromosomes. One is turned off, but the other one is on. And typically that is random. So half of the female cells have one X chromosome on, the other half have it on. And so what you're proposing is a really amazing experiment where you're going to try to turn off the X chromosome that carries the pathogenic variant and turn on the one that has the normal one. That is conceptually feasible. I don't know if anyone's really thinking about, I'm sure someone's thinking about this for other X-linked disorders. But it is an interesting idea.
Okay, thank you.
And Jay, I have a couple more questions here. It's and apologies for it. It can be a very basic question. If we can damage a gene and create a OGT damaged gene,
yep.
Why we all know our mutations? Why can't we just reverse it and use the same technology to just deliver the right copy and change it?
Yeah, so you can do that. It is being done. There was a very high-profile report last summer that came from CHOP, Children's Hospital of Philadelphia, where they did exactly that, where they identified the pathogenic variant, generated a CRISPR reagent, went in, repaired those cells, and they saw some effect. The challenges with that is it really depends on the disease, because we would have to, in this case, deliver the CRISPR to the right place in the body, have it work well in a large number of cells inside the body, and then also assume or hope that that repair of that variant would also either, at a minimum, halt or maybe even reverse the disease. So it is feasible, and that is an application in gene therapy, genome editing-based genome editing, that is really being explored by a lot of people in a lot of different places. The challenges are the details. The devil's in the details of how the CRISPR gets delivered, how it gets to the right cell type, what is the right cell type for a particular disease, and how efficient that can be done. That's why it's still very much an exploratory idea, but one that is exciting.
Makes sense. Thank you. Miranda?
Well, and just another question going off of his question. Is there also a chance that the gene that's turned off is also damaged, or is it always that that's a non-damaged gene and then the other X is definitely the damaged gene?
It would the hope or the assumption is that the undamaged allele is functional. That can be confirmed at least to a first approximation by sequencing. So, in fact, probably for those of you that have daughters that have this disorder, if you've done genomic testing, they have the sequence from the other X chromosome. So that would be known, and it's unlikely that the other OGT or gene that's on the quiet X chromosome is also damaged. It would be very unlikely, but you would have that genetic information available if you've done genome testing.
Yeah, we actually did do the exon activation, and at least my gene it's 91% turned off. So then I guess 9% of that gene is still turned on. I don't think I'm understanding everything exactly right.
Yeah. So what it is, it's not so much. It's that 9% is the percentage of cells that have the correct gene turned on, or vice versa. I apologize. So this comes into an exon activation question that we have to review the details of what that report said.
Okay, sorry. I'm definitely not understanding the 19% and the 9% and exactly what it means. Thank you, though.
Okay. Apologize. I didn't go into a lot of exon activation in this presentation. I apologize. Other questions?
No, I think we're good. Thanks,
Okay.
Jay.
Okay, so the decision experimentally for us to use patient-derived cells versus engineering cells to study a particular disease really comes down to what's available, how quickly we want to get things going, what the resources are available. So I've kind of listed some of the advantages and disadvantages for this. We can go over some of these. So obviously, if we get patient-derived cells and get cells from several different patients and make iPS cells from several different patients, there are some advantages. We can do comparisons between the different cell lines. This is actually very valuable and really essential if there are diseases in which we don't know what the causative gene is. Now, in the case of OGT, we know what that gene is. But for many diseases that are undiagnosed, we don't know what the causative gene is. This is really the only route to take. The disadvantages with using patient-derived cells, obviously you need a group of individuals that are willing to provide cell samples and get them into a laboratory. There's a lot of paperwork, IRB approval and things like that to do. And so it can be slow to develop early on. The reprogramming process can be slow and somewhat expensive, although there's been a lot of progress in speeding that up. And then I mentioned before is because we're isolating cells and generating pluripotent stem cells from each individual, there's a lot of experimental noise in that. Now, that's not insurmountable, but it generally means you need more cell lines from as many individuals as practical. And that can drive up the cost to be able to accommodate that intrinsic experiment or that noise that we have associated with our genetic differences that we all have. Now, in contrast, if we were to engineer the cells to carry these mutations, there are some advantages because the cells are otherwise in an experimental model that is otherwise identical. Conceptually, it can be faster to make these if you know what you're doing with CRISPR. But there are some disadvantages to it. So if there are other genes that may be modifying the disease, which is very interesting and very important to know, engineered cells may not capture that. And then obviously you got to know what you're doing if you're going to use genome editing in iPS cells, because it is kind of a specialized technique. So I think the decision for using patient-derived cohorts of cells versus engineered cells kind of comes down to really what resources and where an individual program is by way of identifying individuals with the disease and capturing those cells. Okay.
Mhm.
All right, a couple of little other features I've just listed here. I'm not going to go into a lot of details because I've covered a few of these, but pluripotent stem cells, there's just a lot of very advantageous biochemical or molecular or cellular features of the cells. So I tried to translate this down to keep it simple. Obviously, we're capturing the genetic material of the patient, and that is for patient-derived cells, and that's really powerful. We can grow very large numbers of these cells. It just kind of comes down to how much money we have, and we can grow dishes upon dishes of these cells. They can grow very readily, and they can grow forever, indefinitely. They're otherwise normal cells. So that's in contrast to other cell types, particularly if you're using cancer-derived cell lines or things like that. Those are cell lines that their genome's all messed up because they're cancer cells. That's what cancer does. They have messed up genomes. Our pluripotent stem cells are normal. So they have the same number of chromosomes, same number of genes as what we have in our body. And so we're looking at an otherwise normal cell that has not been altered experimentally or due to cancer. I talked about the differentiation and the genetic manipulability. So a few other features that really make these powerful tools for disease models. Okay. So I want to give you an example of another utility for pluripotent stem cells that we're exploring and I think is very powerful. And that is, how do we use iPS cells in a dish to explore potential therapies? And so you can conceive that, you can grow the cells in the dish, and there are specialized dishes. I've actually got some right here. I don't know if you guys can see these. These are little dishes. These are called multi-well plates. So we can grow these dishes in these small little wells, and we can conceptually, if we wanted to do, say, a drug screen, we can grow our cells in these dishes, and each one of these little wells, we could put in a different drug. And so that allows us to treat the cells with a large number of different drugs and say, well, what does the drug do? Does it do something that we think can improve the biology of the defects, the deficits associated with this disease? And we've done this with several different strategies, including looking at small drug libraries of, say, 100 different drugs. We're also looking at panels of FDA-approved compounds. There's only about 3,000 FDA-approved small molecules. And so it is manageable to screen under the right conditions and the right methodologies to screen all of the existing FDA-approved drugs. Well, why is that useful? That is called, what we're doing is we're trying to explore drugs that could be repurposed for this particular rare disease. And that's called repurposing. We're taking a drug that has an approved functionality that's FDA approved, but we're going to use it for a different indication or a different disease. That allows us to much more quickly move things into clinical trials and into patients. The other exciting thing is to do novel compounds. So we could screen hundreds of thousands of drugs with this strategy and say, well, that's a discovery. How do we discover new drugs that could be used for these therapies? So because these cells are grown in a dish, we have that capacity to do these types of drug analyses in a somewhat high throughput fashion. Let me give you an example of how we're doing it. And this is not in OGT. This is in another rare neurological disorder. We were able to take advantage of the fact we could genetically modify our pluripotent stem cells, our patient-derived cells, to express a protein that allows us to detect how active a particular biochemical pathway is. And the way this works, we actually the cells glow light when I engineer them in such a way with this transgene. And the transgene is actually derived from the protein that makes lightning bugs glow. And so what we are doing is we're taking that protein and we are modifying it so that way we can track how active that biochemical pathway is. And what we're looking for is, well, which drugs can make that biochemical pathway better? So we essentially do that by looking at how much light is emitted by these cells. And so we put these cells, differentiate them to neurons or differentiate them to cardiomyocytes, put them into these dishes and ask, well, which drugs can make the defective biochemical pathway better? And this is an example of some of our data. Each one of these dots represents a different drug, and you can see how much light is being made by the cells in response to each one of these drugs. And we're very interested in these drugs here that can make that defective pathway better. And so now we have some very exciting drug hits for this particular disease. And this is a small panel of subset of FDA approved drugs. And we are now taking those candidate drugs and putting them into our mouse models of our disease to say, can these drugs, which we identify in a dish, can show some improvement in our animal models. So that's one example in which how we could potentially use pluripotent stem cells in a dish to discover drugs or other therapies for a particular rare disease. Any questions on that? Okay.
Nope.
No question, but I think that it's really awesome what you're doing, like what you're describing here. It's just finding using the tools that you guys have to find drugs that actually work against these diseases. I think that's amazing.
Yeah, well, thank you for that. And I think this is the key for, this is obviously we came up with this strategy for this particular rare disease. We need to think creatively of how we can apply this or some sort of other related screening strategy for something like OGT. And this is where, as we learn more about OGT, what the deficits are associated with these OGT patient-derived cells or engineered cells, can give us hints, and Chad's expertise with knowing the biochemistry of OGT, I think we could think creatively about how do we tailor a particular screening strategy for this specific disease. And I think that's an exciting thing, is how do we apply what we know, but one that is amenable for for OGT. And so this is sort of reiterating, this is the generic pipeline that we just take our cells, look for drug hits, confirm these things in a dish, and then ultimately, I am still a mouse geneticist, and I still believe strongly that being able to do these confirmations in a whole animal model are still very critical. And so this is the pipeline we have taken for a couple of our rare neurological disorders. Okay, so I've tried to touch on a few different things on how we use iPS cells for modeling rare disease. I think a lot of what we're looking at or thinking about with OGT is really kind of firmly grounded in our need for basic research. In other words, our need to really understand the molecular and cellular deficits associated with the disease, and that will then lead us into some of these other areas where we can leverage iPS cells for drug development. Is there a potential for cell therapy? I don't know. Metabolomics, biobanking. I think ultimately we want to be able to make a large number of cells that are available for other researchers who may be more clever than I am to think of new ways to address the disorder. So so I think we're very much in the early phase, I think, with OGT, but I really appreciate the timeline and the goals and milestones you guys are thinking about for your disorder. For all the promise of iPS cells, we always have to be realistic about the challenges. They provide a lot of powerful elements for us to study diseases in a dish. But you got to keep in mind, these are simplified models. These diseases grown in a dish. They aren't children out on the playground doing what children and adults like to do. Organoids are powerful, but they don't mimic the entire organ, and we got to be aware of that. And you have to understand if you want to apply these models to a particular disease that it's being done in an appropriate way. I still believe animal models appropriately generated and tested and well designed are absolutely essential as part of the pipeline. They need not be genetically engineered mice, but that is certainly what we are doing. And iPS cells are they get kind of expensive after a while, especially if you're going to do things rigorously. If you want to look at many cell lines, validate our findings rigorously. So in other words, what we find is real and reproducible. It is not a cheap endeavor, and nothing in science ever is, unfortunately. So that's just the way we're at. So anyway,
sorry to
Yeah.
interrupt you. I was just curious, because I know you work at a children's hospital. Are these cells that are already being used for treatment in medulloblastoma?
we
Are
are
you guys
not.
already doing this for medullo or no?
We're not doing anything with medulloblastoma. I got a guy 2 doors down that is studying medulloblastoma. He's doing it in cell lines and actually also in an animal model, which is actually a chicken model, a chick embryo model. We are studying. We're using iPS cells to study some pediatric leukemias for some interesting reasons that I'd love to talk about, but it's probably beyond the bounds of this discussion. So it is an interesting application. How do we use pluripotent stem cells to study cancer? And the answer is it is possible with some cancers. So we're not. I need to talk to the lab down the hallway and see if they could move into iPS cells for some of their medulloblastoma. But we're not working on it, but maybe other people could.
Yeah, I was just curious. My other son had well, he had different types of medulloblastoma, and then what we found out later was that it does run on the OGT, and he had symptoms of the OGT-CDG as well. So we were trying to see if there was a connection to that, but he passed away 10 years ago. So things have come a long way since then. It's just it's a question that's in my head anyway.
I think it's an interesting question. And Chad, in terms of the link of OGT to medulloblastoma, that's something I'd have to do some reading on.
Sorry.
Well, right now, there's no good links with OGT and cancer. It's elevated in some, down a little bit in others. Cancer cells don't usually want to do anything to their OGT, because then the cell will usually die. In this aspect, though, this is probably one of the cases where that mutation probably tweaked, rewired some protein-protein interactions and some cellular networks that probably promoted that medulloblastoma.
That's what I think too.
It certainly didn't help. And there it's, these mutations, since you still have OGT and you still have GLITNAC, then what you're really looking at is now you've got some potential gain of functions, which is a new interactions or new proteins that are glycosylated that a cancer might find advantageous.
Yeah. Thank you.
Interesting. Okay, so I hope I've given you a little bit of a feel for how we use iPS cells, how they're applied, their utility, but also their limitations. And I will say I don't believe that any one disease model will be sufficient to solve all of our rare disease challenges. I believe iPS cells are a tool in our toolbox that we can apply, but really should be used in addition to some other orthogonal or other additional tools. So I spent 20 minutes this morning with AI on making this little diagram. So I think this is kind of cute to really represent the need that our toolbox has to apply a lot of different disease models and research models to be able to answer the question. And the choice of which one of these different disease models we use from our toolbox will depend on the disease. So I really thank you for everybody for your time, and happy to answer some additional questions. Andre, I know we tried to touch on some things that you had mentioned, but happy to follow up on some additional more details that we could discuss here.
Yeah, absolutely. Hey, Jay, thank you so much for putting that together. See how much excitement it causes in the community. Questions that I have are what is the timeline to produce this cells? That's one, and what is the cost? You mentioned that it's not cheap. What is the cost if we would like to, like for example, fund some of those some of this research?
Yeah.
Um,
So
just a ballparks.
yeah. I mean, so like I said, there's two ways we could make these cells, either patient derived or engineer them. Engineering them, I think we're trying to we're working on the budget right now. Actually, the budget's out there for our grant application. We're trying to envision making the cells, each one genetically engineer them. I'm not a core service, so I'm trying to tally this up without turning it into a commodity. But it's somewhere between maybe $5,000 to $10,000 per cell line, very, very roughly. And so that's the cost. In terms of the timeline, it's a couple months, and that's where we go from looking at the genome, taking a look at the sequences, design, validate CRISPR reagents, make the mutation, get the cell lines validated. That actually takes some time. We got to make sure that the cells aren't weird or wonky, that they didn't pick up anything weird during the course of the genetic manipulation. But it's a couple months to make them. Obviously, it doesn't scale. There is some economy of scale to a limit. I've got a relatively small lab, so I can't throw 8 different OGT mutations on Priyanka all at once. So it would. Our hope is what we're budgeting. I think in terms of our timeline, be able to get these cells made, hopefully in the first year of a 2-year grant, with some aggressive funding and dedicated support for staff and reagents. So, 5 to 10 grand per cell line, a couple months per cell line. If that gives you a really rough, back of an envelope calculation.
Yeah, and what about the patient derived? Is it the same ballpark for both cost and the timeline?
It's a little bit more expensive and a little slower. However, the biggest challenge are the front end logistics for that. So in other words, before we can get one sample from any of the patients, because that is human subjects research, we have to get permission through an institutional review board to do that work. We have to develop and present a protocol to do this work, because we're taking samples from a patient and putting them into an experimental procedure. And so it's as much of the logistics on the front end that slow things like that down. So I would say, 6 months plus that similar timeline that I just discussed, plus maybe 25%, because they are a little bit slower and a little bit more expensive to make.
Sage, go ahead.
So you were saying that it would be more expensive to do the patient derived than it would be to do the other version, like you were saying.
A little bit,
Mm-hmm.
but it's certainly slower, mostly because we need to develop an IRB to get permission to do these things.
Okay,
for OGT.
That actually makes sense, because I know when you guys were talking in the the last meeting that everybody had, talking again about getting samples from patients. I had an appointment with Dr. Morava in between this meeting and the last meeting, so just a few days ago with her, and did speak to her about that, because I am in the process right now of doing other genetic testing with U of M, not only for myself, for my own OGT mutation and exon activation, but also for my 2 sons too. And so Dr. Morava and I were discussing, whatever things you guys are, doing there and asking for patient samples tissue wise. She did suggest to me potentially, if we're already seeing them for a medical reason, asking our doctors if they might take these cell samples and freeze them for the while until you guys have gotten this stuff figured out. Would that be a good option for some of us to be able to do that right now while you guys are waiting?
We would have to look and see what you got to kind of set these things up on the front end before you collect any samples. And so if your clinician or physician scientist that you're interacting with has an institutional review board approval to collect tissues for research purposes, then great, by all means, collect them. Now, the devil's in the details. They got to be sure they're being collected properly, and they got to be frozen properly, because we have to have live cells. Like many times clinicians, this is an overgeneralization, I apologize. Oftentimes when they're thinking about collecting samples, they want to collect samples and just throw them in a fixative or throw them in the freezer. That'll kill the cells. We want the cells to be alive. So the method in which the cells are collected and appropriately frozen and cryopreserved is very important. And all that gets kind of laid out in an IRB. And also whatever institutional review board approval needs to include verbiage to make clear that, you know, these samples can be used for culturing cells longitudinally to propagate. Some IRBs don't have that necessarily, particularly older protocols, in which case that can be problematic. So I would just be hesitant to tell them to capture a sample now without knowing what IRB protocol that's going to be covered under.
Okay, that absolutely makes sense, because I was concerned about going ahead and just pulling these samples as well too. Right now with, you know, the freezing process, I figured there might be, you know, some sort of like cell loss or anything, like, you know, when you freeze food and it loses nutrition. I figured, you know, you guys might be seeing some of the same issues there with that. Now, when you're saying with that, it's the IRB, the IRBs with that, is there like, because you were saying to go with our like institution and their IRBs now, is that something that's like, we would be going institution to institution, or like, how does that kind of move forward? Sorry if you guys discussed this earlier on. I did come in halfway through the meeting.
Yeah. We have that. There's different ways to approach, you know, protocol development for review purposes. So obviously you can do it institution by institution. That becomes inefficient and a little bit duplicative. Another route, so I've worked with other rare disease, you know, boards, is to either, you know, as a foundation, work with a single individual at a academic institution to develop their IRB, or develop a blanket IRB with someone who can help support that. I know you guys have had some discussions with Combined Brain. That is one of the groups that can really accelerate these things, because they've got a protocol that will work for just about anything. You just got to, you know, pay in to play and be part of that. So there are a couple different strategies you can take with that. But the key thing is, before you collect sample number one, that sample number one's got to be appropriately covered under an approved protocol and reviewed and approved by an institutional review board.
Okay, thank you so much for that.
Okay.
Neil? Oh, Neil, hold on. I'll need to unmute you. Or can you unmute yourself? I heard the echo, so I muted you, Neil. Sorry.
At birth, we had the stem cells, frozen, collected, whatever, and been paying the annual fees since then. Would that help in this instance at this point?
It is these are cord blood stem cells.
Yeah.
Probably. What we'd have to do is look at when we discussed other individuals that have cord blood cells, we just got to look and see how many cells they froze away. We need a minimum number of cells to make the reprogramming successful. So we'd have to go back and look. Second off, there are some of those cord blood repositories do not like or will not participate in research studies with their samples. I believe it's a liability issue, or I'm not real sure what their hesitancy is, but I can kind of understand. So we'd have to look and see how they were frozen, how many cells were frozen, and whether or not that particular bank will basically, you signed a contract probably when you sign those over and pay.
Yeah.
There may be a clause in there that says not to be used for research purposes, or can be used for certain purposes. So that's just got to be clarified. It gets into the nuances of legal law stuff.
Yeah.
That I am wholly unqualified to bring up, but it is feasible. They can be very valuable, but we just got to see what the details are on that.
Okay, so it's better to have it and figure out the details from there.
Well, what you got is what you got. It's in the freezer, and then we just have to look and see what is allowable.
Okay, great. Thank you.
But Jay, I think this, we also have stem cells. I see that Sage pinged the chat group that she also has for boys. The question is, how is it different from patient-derived cells? Looks like you don't need those cells like those stem cells that we got from birth. Looks like you just need a blood sample or tissue and have a patient-derived cell. So do we even need to talk about that? We keep bringing these things. All of our parents, myself included, we're just like, oh, we got the stem cells. Do we need them? And researchers just say, look, well, some of the researchers say, well, we kind of don't need that anymore because we can do iPSC from any tissue now.
That is correct. So with the exception of an individual who's passed away, it's not hard to collect a blood sample, and those work just as well. It may work better because you can collect potentially more blood from an individual and purify it in such a way to make it. So the cord blood cells, I think, still have some utility, but for this iPS cell generation, it is less relevant anymore. Correct.
Okay. Yeah, makes sense. So Neil, Sage, myself, looks like we don't have to be super excited about our cells and whatever we're paying to those biobanks. And it may be really more beneficial to just go and ask Eva Morava to pass the blood samples. But this is something that we need to do. Sage, we need to reach back to Eva and ask, hey, how do you store those cells? If we arrange with Jay that Jay will produce, his lab produces the patient-derived iPSCs for us, will it be potentially possible? Like do they are they stored in the right way? So this is something that we need to definitely start doing. Another big call out here, guys, that the patient-derived cells, it's a paid service, right? So it's something that we'll have to fund ourselves somehow. And we don't know the price yet, but Jay, if you could just share the price with us at some point, that would be great, so that we could just maybe like centralize the IRB plus this work together, because we were talking about the induced cell lines, right? Induced cell lines. No, hold on. The isogenic background, and this is something that Jay and Chad are working on a grant for. And there's a potential for this grant to be approved, and in this case, they will be producing several cell lines there. But patient-derived are not covered by that grant, and we may want to fund it ourselves, maybe on an individual level. Guys, I'm definitely looking forward to it. I'll ask Jay to provide the quote for that, and we can connect Eva and Jay and see how this can happen.
Yeah, happy to further that conversation.
Yeah. So, and another thing that I was researching, just again for the community, that isogenic background cells they have like the same genetics background, but just different OGT mutation of our kids. And it is very good to study the disease at a broad level, just to find what are the commonalities across different variants. But there's additional value to have our own patient's cells, because it provides the whole entity of the genetics background for a patient. Because maybe there is not just OGT mutation that is causing the intellectual disability or other things, but there could be something else that isogenic cells may not show. So looks like it has to be like both things that we need to work on. Sage, go ahead.
This is less so a question for researchers, but more so for the other parents here, because I was having this discussion in the comments on our page the other day. I was interested with you mentioning that about the intellectual disability, Andre, because I'm wondering how many of the kids or anybody else here has gotten the kids to have a comprehensive, like a neuropsychological and developmental eval, and actually had that testing to see in the exact areas of where the delays are in their brain. Because I know for myself, for that testing, they told me I had various cognitive delays, but that I don't have an IQ delay. And I think now, as the boys are getting a little bit older, and now that they are verbal, I think that they've had a lot more neurological problems that were impacting things that made it seem like they were more intellectually delayed than they were previously. So I'm wondering if anybody else has gotten that more specific testing done for their kids and seen like the exact areas of delays that they've had going on.
Yeah, we just had one, Sage, in December. We've gone through a very extensive specialized assessment, which showed that our like almost 6-year-old Victoria, she's like one, like they say that like less than 2 years old, like equivalent of development. So we had that, and we have like the full report, which basically states that she's like severely affected.
Okay, yeah, and I was wondering about that because I know, like, for my profile and it's now again, like I said, seeming for my kids, we have a lot more motor issues and delays, and like I said, they all seem to be more neurological and muscle based in my family, especially because now my mother has also tested positive and she has stroke-like episodes, and you know she does have like oh goodness, I can't even think of the word right now. Thank you for my own aphasia. Dyslexia. She has dyslexia and such. And you know, so she was always a little bit slower growing up in school too. But you know, I'm just goodness, I was just, like I said, I was just wondering to see if everybody's been doing those tests and if they've been doing them regularly to track that progress too, because I know my son Zachary has been doing those regularly since he was about 2 years old to kind of keep tracking that developmental like progress with the brain and everything.
Miranda.
I'm just going off of what Sage is saying. Yeah, I think that that's a good thing for a lot of our families to do is do the neuropsych evals or any other testing that we've had done to follow progress to make sure there isn't regression, and just to make sure they are progressing.
I mean, our experience is our daughter had infantile spasms at 6 months. So Dr. Morava said she expected intellectually higher level than what she presented physically. So she may be impacted by that, but intellectually she's quite impaired. Basically non-functional at all.
It is interesting how, depending on where all of our variants fell, how the symptoms are different for every child.
Mm-hmm.
And that is, and I put this in the chat, this is where a natural history study is going to be very important. And so I hope you're interacting with some clinicians that are taking a truly deep dive into this. And what you bring up, Miranda, is what we call in the field of phenotype-genotype correlations. So, you know, are these, you know, variabilities in disease manifestations due to the different variants or different mutations, where they occur, what the biochemical deficits are, yeah, but also what other things can impact that, as Andrea brought up, other these other modifiers. All that is absolutely essential to, you know, document in a really detailed natural history study. And so it's something, you know, I hope your foundation is really thinking about and finding the right clinical collaborators to have that available. The FDA will absolutely require that before they move it into anything in terms of a therapeutic development.
A previous conversation on that call exactly 2 weeks ago was with Marie from Dan's lab, who's dedicated to run natural history studies. So she's they're working on two things: natural history study and biomarker discovery. So yeah, she's on the mission to run it and complete it.
Fantastic.
All right, we're at time. Jay, Chad, appreciate you being here, and even more so appreciate your initiative to basically initiate the grant, right, which will hopefully allow us to get those iPSCs. We'll be closely following your progress, and if you have any other topics that you could bring to this group, we'd be happy to have you here. And again, thank you as well. Thank you. Thanks again.